Could you ever imagine a world with no diseases? How about pigs that can fly? How about babies with guaranteed perfect hair, body, and brain, unlike the most of us? Well, for the first time in history, all of them might just be in reach. And we swear we're not trying to scam you out of anything. All of those can be accomplished with two simple words: genetic engineering. Genetic engineering is defined as the process of altering the genetic makeup of an organism by changing the sequence of a gene, adding a new gene, or deleting a new gene in the DNA sequence. For many millennia, the only way for organisms to change their appearances or their abilities was via natural selection and random mutation. Over many years, organisms develop traits that are beneficial to their survival, and this is carried on to their next generation. But humans have always found a way to tinker with the natural order, and even all the way since the 1960s, numerous tests have progressed the process so much that by 1972, rDNA, or recombinant DNA that has been created via the combination of elements of DNA from different organisms, was first created by Stanley N. Cohen and Herbert W. Boyer. By 1981, Thomas Wagner at Ohio University successfully transferred a gene from a rabbit into a mouse genome, creating the first instance of DNA microinjection. Literally the next year, Genentech scientist Dennis Clade managed to genetically engineer insulin, effectively eliminating the required slaughtering of animals for their pancreas just for little bits of insulin. In 1988, the first genetically modified crop, known as BT corn, was officially introduced to the US market, and this crop was genetically engineered to be resistant to pests and some viruses, simultaneously increasing yields. Finally, in 1993, Francisco Mojica discovered that some DNA in bacteria were being repeated, unknowingly discovering the basics of CRISPR, or clustered regularly interspaced short palindromic repeats, when he was observing the DNA sequences in bacteria. For the next 10 years, he continued to observe the repeats in many DNA sequences, until 2003, when the repeating DNA he was observing in bacteria matched with the DNA that viruses have as they attack the bacteria. This crucial finding led to the ultimate establishment of CRISPR, and where our topic actually starts.